I'm pleased and flattered to have been invited to give this address at the conference this year on mesothelioma. To my sincere regret, I can't be there, but it is indeed a luxury to be able to talk with you at such great distance on the biology of mesothelioma, at least from one perspective, and on the possible novel ways of treating it. Mesothelioma is, as you know, a very unusual disease, but increasingly we appreciate the disease as having many affinities to other types of epithelial tumors, that is, carcinomas, which in aggregate constitute, as you also know, about 80% of clinical burden. In the case of almost all of these tumors, secondary tumors, that is, the metastases, represent the great source, the great causes of mortality in the oncology clinic. Here, I'd like to show you in the first slide the fact that we kind of understand the first steps of tumor progression, at least in the context of the colon, as first laid out by Vogelstein in 1989. And I want to focus not on the formation of the primary tumor, which is here indicated as carcinoma, but rather on the last step, which is the process of invasion and metastasis that creates the metastases that are important ultimately for about 90% of cancer-associated mortality. This last step, going from a primary tumor, a carcinoma, to metastases, itself subsumes a whole series of sub-steps that in aggregate are called the invasion metastasis cascade. And here you see them laid out in the lower right of the slide, involving localized invasion, intravasation into the lumina of microvessels, translocation through hematogenous routes, extravasation, and ultimately the invasion into the parenchyma of distant tissues, where these cancer cells are able to form initially micrometastases and with low efficiency, fortunately, macroscopic metastases that become clinically apparent and ultimately indeed life-threatening. Having focused on that, I would mention that we're confronted with the issue of how cancer cells are able to acquire all of these various capabilities that are implicit in passage through the invasion metastasis cascade. That is, how can cancer cells negotiate these complex steps? And one answer, which may indeed be the dominant answer, is that they can activate a cell biological program that is termed the epithelial mesenchymal transition, and the EMT, which is known to reside in a latent form in many epithelial tissues, and which is also exploited opportunistically by cancer cells as they become highly malignant. And here you can see one image which indicates that in a very dramatic way. We see here that the carcinoma cells are stained with a keratin, which is a epithelial marker. Surrounding them at a distance are the blue nuclei of recruited stromal cells, and the critical partners in this process here are indicated by the green cells. These are carcinoma cells of human origin. This is, after all, a xenograft that have shut down the epithelial marker cytokeratin and instead express the mesenchymal marker vimentin, indicating that these cells have gone from an epithelial to a mesenchymal state, that is to say, they've passed through the EMT through activation of their previously latent EMT program. And this program confers on these cells not only a heightened resistance to apoptosis, but also motility, invasiveness, and indeed imparts to them many of the capabilities that they require in order to physically disseminate from the primary tumor and thereby seed secondary malignancies at distant sites. If one were to summarize the multiple benefits conferred on the tumor by the EMT process, by activation of this previously silent cell biological program, one could summarize them in the following ways. First, physical dissemination is enabled when carcinoma cells in a primary tumor activate this program. Secondly, this imparts to these cells the ability to self-renew, that is, to function as stem cells, in that cells that have passed at least partially through an EMT often acquire the ability to become cancer stem cells that enables them in turn to serve as the founders of new tumor colonies. And that plays a particularly important role in the process of metastatic dissemination, in that the disseminated cells must be capable of seeding new metastatic colonies. Finally, as it turns out, passage through the epithelial mesenchymal transition, at least partially, going from an epithelial to mesenchymal state, heightens the anti-apoptotic responses of cells and thereby confers on carcinoma cells a heightened resistance to many forms of conventionally available chemotherapeutics. And with that last point in mind, one can refer here to the third slide, in which is indicated a hypothetical scheme of what may happen in many kinds of human tumors where the stem cells, the cancer stem cells, are depicted in gray, and the more differentiated progeny, which constitute the bulk of the cellularity in the tumor, are depicted in pink. And here we see the fact that as a consequence of the cancer stem cells having an elevated resistance to various forms of conventional therapy, they tend to persist after the initial onslaught of conventional therapy, either chemo or radiotherapy. And therefore, initially after treatment, the cancer stem cells are there, albeit in very reduced numbers, but because of their tumor initiating capability, are empowered with the ability, after therapy is halted, to regenerate new tumors, as you see on the right of that slide. And that indicates that the cancer stem cells are true enemies in two senses. First, because of their heightened resistance to many forms of existing therapy, and second, because of the fact that they also happen to harbor tumor-initiating capability that in turn empowers them to form new secondary tumors. And so, therefore, this provokes the question of whether one can actually develop therapies that can preferentially eliminate the cancer stem cells, admitting for the moment that we already have therapies that target the non-stem cells within tumors. And that indeed is one of the agenda items of our work, and beyond that, the work of Verastem, whose work I will present today. Here you can see one indication of the fact that conventional chemotherapeutics, in this case the use of doxorubicin and paclitaxel, are able to eliminate the non-stem cells from a tumor at lower drug concentrations than the stem cells. In these two graphs, the non-stem cells are in blue, and you see that they are eliminated, that is, their viability is reduced, as indicated on the ordinate, but by drugs at a 6 to 8-fold lower drug concentration than are the stem cells, which are indicated in both cases in red. And this is true both for paclitaxel, and it's true for doxorubicin, and indeed one believes that this holds true also and extends to a variety of other conventional cytotoxic therapies that rely, in all cases, on the ability to trigger the apoptotic response in cancers and thereby eliminate the bulk of tumor cells. Having said this, we've recently learned some of the dynamics at the molecular level that enable cells to enter into the cancer stem cell state and to persist in that state. And interestingly, one of the important characteristics of this, which also enables cancer cells to metastasize, given the linkage between metastasis and tumor-initiating capability that I described before, is the ability of cells that have passed through the EMT, the epithelial-mesenchymal transition, to extend long finger-like processes that we call Filopodia-like processes. You see images here, and it turns out that cancer cells that are able to disseminate from the primary tumor but are unable to extend these processes are stuck once they extravasate into the parenchyma of distant tissues. The cancer cells that extend these Filopodia-like processes are able indeed to form linkages, stable anchorages with the nearby extracellular matrix of the parenchyma of the host tissue, for example the lung, and thereby able to activate a series of signaling cascades which culminates in the activation of focal adhesion kinase and the ability of the recently extravasated cells, once they've done so, to begin to proliferate. Conversely, non-metastatic cells fail to extend these Filopodia-like protrusions, which are covered with integrins that in turn enable the physical anchorage to the extracellular matrix of the parenchyma of the tissue into which these cells have extravasated. And lacking that ability, these cells are unable to activate focal adhesion kinase, and thus their mitotic program is effectively aborted. They just get stuck there and represent latent cells, dormant cells, that may never be able to do anything. And therefore the extension of these processes is really critical to the post-extravasation proliferation of disseminated cancer cells. Note, please, here the critical central role of focal adhesion kinase in this process, in that in the absence of focal adhesion kinase, the extravasated cancer cells are unable to proliferate, and as I'll mention shortly, this is also critical to the acquisition of the tumor-initiating capability of these cancer stem cells. And here you can see, by the way, a schematic cartoon in the upper right of how the integrins that are displayed on the surfaces of these Filopodia-like processes, as they're called, FLPs, they're constituted from alpha and beta integrins. This is the case with all integrin heterodimers, but associated with them in the cytoplasmic tails of these integrants is the focal adhesion kinase that is the gatekeeper for subsequent proliferative or mitogenic response. Indeed, the ability to form mature focal adhesions, which you can see in the lower right of the immunofluorescent micrograms here, the ability to form these mature focal adhesions represents a critical rate-limiting event to the ability of these cells, once they've extravasated, to proliferate, and, as I'll mention shortly, the ability of these cells to function as tumor-initiating cells, that is to say, cancer stem cells. Here we see the preferential effects of VS6063, the Veristem drug, on cancer stem cells. And in the top left graph, we see the fact that if cancer stem cells, populations containing both cancer stem cells and non-stem cells are treated with VS6063, then the population of cancer stem cells is gauged by the use of the Alder-Fluor marker for cancer stem cells. The population or the representation of cancer stem cells in the blue curve declines precipitously, whereas the overall size of the population as a whole in the red curve is hardly affected at all. And that begins to persuade one that VS6063 is indeed preferentially targeting the cancer stem cells. The central role of focal adhesion kinase in the ability of the cancer stem cells to reside continuously in the cancer stem cell state is revealed in part by the top right histogram, in which one sees that by knocking down focal adhesion kinase, one significantly decreases the representation of the Alder-Fluor positive cancer stem cells in this cell population. And once again, one gets an indication of preferential targeting, albeit using a different criteria. In the lower left, we see two histograms that provide even more rigorous, to my mind, demonstration of this. And in this case, one was allowing tumors to grow out a bit, treating them as the tumors were growing in vivo with VS6063, and then, after the tumors had been treated, extracting the tumors and the tumor cells and reimplanting them back into mice, asking the question about their residual tumor-initiating capability. And as you can see, in the case, in the left histogram, you can see that the treatment with VS6063 resulted in perhaps an 80 or 85% decrease in the tumor-initiating rate of the cancer stem cells that had previously experienced VS6063 in vivo when they were reimplanted in a new set of mice. Conversely, if one treated mice in parallel with paclitaxel, which obviously targets the non-cancer stem cells, here one sees that the relative representation of the cancer stem cells is increased enormously, as you can see from the red bar on the histogram. So these together begin to converge on the notion that VS6063 preferentially targets the cancer stem cells, and it does so in part through its ability to somehow disrupt the program of stemness, which in turn is dependent, as I've argued before, on the intermediation by focal adhesion kinase. In the next graph, which we see here in the next slide, what we're looking at now is the ability to treat cancer cells in vitro, in this case mesothelioma cells. You'll be relieved to hear we're finally talking about mesothelioma cells, either in vitro with either a control buffer or with pemetrexed or with VS6063 or with a combination of VS6063 plus pemetrexed, and then looking at the ability of the treated cell populations to see tumors subsequently in mice. In the graph below, we see the representation of tumor-free mice that resulted from the implantation of these mice, of these cells, these treated cells. And as you can see from the in the control population, in which the mesothelioma cells had been treated ex vivo subsequent to their implantation in vivo, here you see that within 3 or 4 weeks, already half the mice had died from implanting these cells that had been treated previously ex vivo. Mice treated with pemetrexed, they survived a bit longer, as you can see in the red curve, and thus they did indeed receive some benefit from the overall treatment with pemetrexed. But what's most critical and most interesting are the blue and the green curves above, the horizontal curves, which are superimposable and which indicate that effectively treatment with either VS6063 alone or treatment with VS6063 plus pemetrexed effectively eliminates the ability of these ex vivo treated cells to seed life-threatening tumors. in mesothelioma tumors in these host mice. One can extend this even further with a final graph, which I show you here, in which, in fact, one can look at seeding cancer cells as xenografts in patient-derived xenografts of mesothelioma in vivo, and then beginning to treat them after a treatment of conventional chemotherapeutics with either cisplatin, pemetrexed, with a FAK inhibitor. In the blue curve, one sees the outgrowth in terms of tumor volume of these xenograft tumors in which the cells have not been treated at all with either cisplatin, pemetrexed, or the FAK inhibitor. However, as one sees here in the red and the green curves, treating the cells with just cisplatin and pemetrexed, that is the conventional agents which are acting together, extends or really retards the growth of the tumor cells significantly in the green curve. But if one combines these two conventional agents, as we can call them, with a FAK inhibitor, then one gets a much greater retardation in the overall growth of the tumors, which in this case extends over a period of almost 7 or 8 weeks. So here one begins to see that one can anticipate synergies between conventional agents that are already approved for utilization in the clinic, cisplatin, pemetrexed, and novel agents such as the FAK inhibitor, VS6063, which Verastem is now attempting to see can actually confer benefit, clinical benefit in mesothelioma patients, including a long-term, durable clinical response when combined with conventional agents. Having said that, I'd be very pleased to take questions, and indeed some have already been forwarded to me, and I'll try to field them one after another.
Thank you, Dr. Weinberg. On behalf of your colleagues at iMig, we thank you for your time for this presentation. And my name is Tom Prunty, and your colleagues at iMig have submitted questions to me they would like you to address for the benefit of the assembly in Cape Town. So first of our questions for you, Dr. Weinberg, is what's the best way to define a cancer stem cell?
A cancer stem cell is best defined not by the markers that it bears, but operationally by how it behaves functionally. That is to say, if one takes a population of cancer cells and subjects them to various degrees of limiting dilution and implants them in a mouse, to what extent are there cancer stem cells there, as defined directly by the ability to seed the outgrowth of a new tumor? In other words, the operational definition of a cancer stem cell is tumor-initiating capability. There are multiple markers for the presence of cancer stem cells, such as aldofluor, but none is as definitive and specific as the functional operational test I just defined, i.e. tumor-initiating capability.
Thank you. Next question is it true that pemetrexed is killing the tumor cells, but the cancer stem cells can evade this action? And if so, is this a class effect and also applicable for other chemotherapeutic agents like gemcitabine?
It seems to be the case from some of the evidence I presented to you today that in fact pemetrexed is rather unsuccessful in eliminating the cancer stem cells, although to my mind there needs to be more specific and detailed experiments undertaken to prove that definitively. Certainly indirectly one can already deduce that that's likely to be happening because of the synergy between VS6063 and the observed effects of VS6063, as I just showed you before, and pemetrexed. So the question is this a class effect, where one defines class as the class of chemical compounds that are able to act on, for example, mesotheliomas? And here I would say that it is my belief, and I underscore the word belief, that at present the great majority of cytotoxic drugs in a variety of tumors act preferentially to eliminate the non-cancer stem cells, leaving behind or sparing the cancer stem cells, or at least eliminating them relatively ineffectively. It's my belief, and I can't prove it to you at present, that this is not a class effect, where class is defined as a class of chemical compounds or chemical structures.
Thank you. And a question from Dr. Voss. If the cancer stem cells are the cause of regrowth of the tumors, can we apply other anti-cancer treatments like immunotherapy or radiotherapy to kill these cancer stem cells?
In fact, that's a very interesting question that cannot really be answered definitively at present. We do know that cancer stem cells have a heightened anti-apoptotic response, and that would suggest, but it's not really been examined critically, that they'll also exhibit a heightened resistance to to radiotherapy as they have against conventional chemotherapeutics. But again, that's something that needs to be established definitively in an empirical fashion. What about immunotherapy? Well, at present, we haven't even begun to explore the convergence of the rapidly exploding field of immunotherapy with the elimination of cancer stem cells. We don't really know whether the cancer stem cells will express certain kinds of specific antigens that will render them particularly susceptible to elimination by some of the recently developed immunotherapeutic regimens. And so that's a question that represents an unknown at present. I'm not saying they can't, and I'm not saying they can be eliminated. We still need to examine that interface between immunotherapy and cancer stem cell research. It's probably the case that the great majority of conventional chemotherapeutic drugs, as I've mentioned already, probably are relatively weak in eliminating the cancer stem cells relative to their proven efficacy in eliminating the non-cancer stem cells from a tumor.
Thank you. The next question, do you think cancer stem cell targeting can make an impact in poor prognosis cancers?
I believe that cancer stem cell targeting can make an impact on poor prognosis cancers. Much of what we know now would urge one to reach that conclusion, but this is ultimately an empirical issue. Will this actually work? Can one actually show that patients who have been treated with a combination of a cancer stem cell agent, anti-cancer stem cell agent, and a more conventional agent targeting the non-cancer stem cells, will they actually exhibit greatly increased survival, indeed even symptom-free survival, or indeed a durable clinical response? You might say, why don't we just get rid of the cancer stem cells with something like VS6063? But the fact of the matter is, we have evidence that if you have only surviving non-cancer stem cells, they're able to regenerate de novo new cancer stem cells. And that causes me to argue that both populations need to be targeted, each with a specific agent.
Is there anything different about cancer stem cells and cancers of different types?
We don't really understand the biological distinctions and the biochemical distinctions between the cancer stem cells in a variety of different kinds of tumors. It is my conviction, remembering that people who are convinced of things are often wrong in a loud voice, but it's my conviction that in fact the cancer stem cells in a variety of carcinomas will have a similar set of molecular regulators, and that lessons learned from one kind of carcinoma study, for example, the cancer stem cells of the breast, will also prove to be applicable to a variety of other kinds of cancer stem-like cells in carcinomas of the colon or of the lung, or in this case of mesothelioma. That generalization has yet to be proven, but there's already wisps of evidence that in fact there are very common molecular elements in the high-grade tumors from these diverse sets of tumors.
Is stemness intrinsic to a cell or can it be modulated by the tumor microenvironment?
The question is whether the average non-cancer stem cell within a tumor can itself become a cancer stem cell. Is that intrinsic to it? And we know that this conversion can happen, albeit at a low frequency in certain kinds of tumors. We don't really realize with any degree of quantitative precision how often non-stem cells can become stem cells within tumors, but we do know that the contextual signals that carcinoma cells receive from the adjacent recruited stromal cells, the so-called inflamed or activated stroma, the activated stromal cells will often send signals that impinge on the carcinoma cells and actually actively induce them to activate their previously latent EMT program. So indeed, the behavior of cancer cells vis-a-vis the cancer stem cell state, granting the connection between activation of the EMT and entrance into the cancer stem cell program, those dynamics we believe can be actively perturbed and influenced by heterotypic signals that carcinoma cells receive from the nearby or adjacent recruited inflammatory stroma of many kinds of tumors.
Your colleague, Dr. Fennell, would like to know, is stem cell genetic heterogeneity the main barrier to cure with stem cell targeted therapy?
At present, we don't really understand how much genetic heterogeneity there is within populations of cancer stem cells. In general, they represent numerically relatively small minority populations, often being present in 1, 5, or even 10% of the total cellularity of a given tumor. We don't really understand how genetically heterogeneous they are, and we're still far away from knowing whether there are subpopulations within the cancer stem cell compartment, admitting it's already relatively small. There are subpopulations that are actually more resistant to certain kinds of therapy than are the bulk of the cancer stem cells, again reminding us that as a whole the cancer stem cell population is numerically a relatively small population, a subpopulation of the cells within a tumor.
And do you expect that the importance of cancer stem cells is likely to extend broadly through the epithelial cancers?
It is my belief, my conviction, as I said before, that the lessons that I've been describing today are going to eventually extend to all kinds of epithelial cancers. That is, there's already evidence, indirect, sometimes direct, that many of the same rules will apply eventually to all kinds of carcinomas, which are organized on very similar molecular plans and which are likely to have very similar molecular regulators. And therefore, people in my laboratory doing work on, for example, breast cancers largely today, do so with the faith that the lessons we learn with mammary carcinomas will in turn prove to be generalizable to a whole variety, a whole spectrum of carcinomas. If you ask my own personal belief, it's my feeling that eventually one will find that all carcinomas have cancer stem cells within them, that they all have a very similar molecular makeup, and that the differences in behavior of different kinds of carcinoma cells will be a reflection largely of the cell of origin of the tissue in which these carcinomas arose. But that the basic overall molecular plan of these cancer stem cells will be very similar from one carcinoma to another. And if this were true, this would be very heartening, because carcinomas, as we know, constitute roughly about 80% of the fatal cancers that one encounters in the human population.
And do you think the importance of FAC that you've shown in cancer stem cell biology is likely to extend to multiple cancers?
I think it's still early days. I was referring here to FAC, focal adhesion kinase, and the fact that it's proven to be an important intermediary in actually conveying signals from the extracellular milieu, from the extracellular matrix into cells, and that enable cells in turn to proliferate and to, for example, persist in the cancer stem cell state. I cannot say whether this will prove applicable to all kinds of carcinomas, but again I reiterate my conviction that very similar molecular and biochemical rules will ultimately be found to apply to a wide spectrum of different kinds of carcinomas. That remains to be validated empirically, which it is not at this point.
And finally, Dr. Weinberg, do you predict that cancer stem cell targeting, combined appropriately with agents targeting the bulk tumor, will deliver an incremental gain or a monumental leap in the war on cancer?
Ideally, knowing what we know now, we would predict enormous synergies between conventional agents that eliminate the non-cancer stem cells in the tumor, together with agents like VS6063 that preferentially attack the cancer stem cells. And I could imagine that in fact one will get extreme extensions of clinical responses, and that is a durable clinical response. However, that remains to be indicated empirically in the clinic, and at present these are only speculations on my part. Importantly, we don't really know the best way of realizing these synergies. You might think, by the way, that eliminating the cancer stem cells should on its own result in a durable clinical response, but we have reason to believe on the basis of experimental preclinical findings that in fact the non-cancer stem cells in the tumor can spontaneously regenerate de novo new cancer stem cells. And so one really does need to target both subpopulations of cells. Granting that, that raises the question of whether the anti-cancer stem cell agents and the agents that target the non-cancer stem cells should be applied concomitantly or serially. For example, eliminating all the non-cancer stem cells initially and then targeting thereafter the surviving cancer stem cells with an agent like VS6063 to ultimately generate the kind of durable clinical response that we all hope for. At present, again, these are matters that need to be tried out empirically, because there's really no theoretical reason or conceptual reason to think one will be much more effective than the other. We just need to try these things out. Again, I would like to thank you for having me here, and I trust some of what I told you turns out to be interesting for your own work and your own attempts to get under control mesothelioma, which until now have been very difficult to get under control. Thank you.